like to welcome everybody to the MBB Mind, Brain, and Behavior Distinguished Lecture Series. I'm just going to give a very short little history about the lectureship series, and I'll turn it over to the co-director with me of the Mind, Brain, and Behavior program, Ms. Welke, who will introduce Stanislas Dehaene, our speaker for today, and today's. So the Mind, Brain, and Behavior lectureship series have been going on for several years now. And the lectures are designed to bring scholars to the university who can speak to issues that have to do with mind, brain, and behavior. And on the one hand, this might seem like a fairly easy task. On the other hand, it turned out to be quite a difficult task. Choosing the lecturers is difficult for the following reason. The lecture series are typically 3 lectures. So it has to be someone who has a story to tell, not just one story, but multiple stories. They have to be able to build up that story over a period of 3 days. And they have to be able to integrate information on mind, brain, and behavior, which is also not that easy. And then lastly, they have to be good communicators to the public. So that ends up eliminating quite a lot of people. But we've had a great series so far. I just want to list some of the names. Marc McClintock, who spoke very recently, Bill Newsom, Nick Humphreys, Noam Chomsky, K. Reginald Jennison, Tony DiMaggio, and Jean-Pierre Changeux. And it's particularly nice to have Stan Dehaene here for these next 3 days, in part because he has worked so closely with Changeux, and I think you'll see some of the signatures there. So before turning it over to Liz, just a reminder that we're going to have 3 lectures. Today's beginning a little bit early at 4. The next 2 will be at 5 in the same place. And the format for each of the lectures will be that Stan will be talking for about 45 to 50 minutes. There will be then 2 faculty members from different departments commenting on the lecture. Stan will be able to respond to it, and then we'll open it up to questions. So we expect the entire event to be on the order of about an hour or so each meeting. So without further ado, let me turn it to Ms. Welke.
It's a huge pleasure to be introducing Stan Dehaene, but I'm going to be extremely brief, and once he starts to talk, you'll understand why. Any extra minute that I take up and take away from him, you will resent. So let me just tell you really quickly that he's had a very interesting route to his current field. He started his education specializing in mathematics at the École Normale Supérieure. Then he moved into applied math and computer science, and then conducted his PhD work in 2 different fields. He really got 2 different PhDs. The official one was in cognitive science with Jacques Mehler. The unofficial one was in computational modeling, neural network modeling of higher cognitive functions with Jean-Pierre Changeux, as Marc was saying, work he started in graduate school and continues to this day. And then finally, he moved officially to cognitive neuroscience with a postdoc with Mike Posner at University of Oregon. He then became a research director at INSERM, where he funded the Cognitive Neuroimaging Unit that exists to this day. It's officially the same unit, right, although it has now moved from its original home in Orsay to their new center in Saclay, both just outside of Paris. Now, beyond those facts, I have to say that Stan, I think, is about the most intellectually ambitious person working in the field. Now, he's taken on what I think are our field's 3 most important and most difficult questions. What is consciousness, and how does the brain give rise to conscious experience? What gives humans access to abstract entities like number, space, and time? And how does the brain engage in abstract reasoning? And finally, how do biology and culture interact to produce uniquely human cognitive achievements like reading and mathematics? Now, in all of these cases, Stan is certainly not the only person who's asking these questions, but I think he asks them with a rigor that hasn't been seen in the sciences so far. So to give you just one example, in the domain of mathematics, Stan aims for a multi-leveled, mechanistic understanding of human knowledge of number and mathematics, an understanding that's as broad and deep and precise as the understanding that one might hope for low-level perceptual or motor functions in animals in neuroscience. He also aims to specify the computations involved in constructing and operating on number. And there he aims to be as specific and concrete as a computer scientist would be, who would try to actually implement those functions by building something that could do them. Now, like the best neuroscientists, he uses multiple converging lines of research to address any given question. He combines behavioral methods with neuroimaging methods and applies them to creatures as diverse as college sophomores, people with different distinctive patterns of brain damage, human infants, and people from far-flung cultures. Now, Stan's work has been very highly recognized and honored. I'm not going to list all his honors. Let me just mention, in addition to the Collège de France professorship, that he was one of just 2 cognitive scientists to win the McDonald Centennial Fellowship, that he's gotten numerous awards for his landmark book, The Number Sense, that he's been elected to the French Academy of Sciences and has a ton of other prizes and has even been honored by the Pope. Now, at this point, you're probably expecting 2 things from this guy. First, you're expecting someone who's very old, and second, someone who's very serious, right? These talks might even be boring if there's going to be all of this serious, hardcore science in there. Well, neither of these things is true. Stan is actually shockingly young. I think he was 24 when he first became an INSERM investigator, and he's now the youngest member of the Collège de France. He's also extremely fun, and probably the most fun person that I've ever collaborated with on bleary trips to Paris, jet-lagged, barely keeping my eyes open. I encounter Stan with this gleam in his eye and some of the craziest, zaniest ideas about experiments that he could do. So it's a great pleasure to have him here at Harvard. He's going to be giving 3 lectures, roughly, I think, tracking the 3 big questions that his work addresses. Today's lecture focuses on the topic of his forthcoming book on reading, called Recycling the Visual Brain for Reading. Stan.
Well, Liz, I cannot thank you enough for this wonderful introduction. It's such a pleasure to be here, and I would like to acknowledge our collaboration with Marc, collaboration with many other people here, and the sharing of ideas, which is the whole idea of the mind-brain behavioral lectures. It's been an incredible stimulation for me, and I think I will present today ideas that are not just mine, but are those of people in the audience, and especially those of many people in my laboratory. This is not the work of one person, of course not. And especially the collaboration with my neurologist colleague, Laurent Cohen, was crucial in what I'm going to present to you today. 32,000 years ago, men entered the cave at Chauvet, the so-called Chauvet cave, at the Vallon d'Arc in Ardèche, France, and they made these incredibly beautiful paintings that today is the same brain we can recognize as painting of horses and lions, but also some symbols that we don't understand exactly what they meant, like hands, or a little bit more recent in Lascaux, these shapes that may look like symbolic shapes. 5,400 years ago, other men began to design more symbolic ways of depicting their mental states, and especially depicting language, using first different shapes of objects, and then as the writing became more and more abstract, cuneiform writing, hieroglyph writing, and in other parts of the world, similar symbol systems emerged for Chinese and Maya writing. And in parallel, in fact, starting as early as maybe 8,000 years ago or even before, they began to make engravings such as this plate from the Grotte du Taille in France, which has a series of engravings that suggest a kind of counting scheme or arithmetic scheme, and is progressively developed into ever more abstract representations of calculations, systems of geometry, and finally very elaborate systems of mathematics that shape the world of science as we know today. There's been an incredible transformation in the capacity of the human mind with such cultural tools, and the question I want to ask today, as Liz says, is probably too ambitious, and I'm not going to answer it, but I would like to ask what is the reason for this cultural singularity of the human primate. If you give to a young chimpanzee a writing tablet like Tanaka and Matsuzawa did, and you let them play with this tablet, they will produce some graphic depictions, but they are not really representing in any way what the chimpanzee is seeing. They are not either developing complete symbol systems like us, they will just sort of play and draw curves on the tablet. If you give paint to a chimpanzee, they might produce compositions. They can be considered non-random, but they are not at all of the same kind of elaboration that we can see in the Shoei paintings, for instance. There is no comparison. So what is happening here is probably not a matter of not being able to learn some of these skills, because with intensive training in the laboratory, animals can learn to use tools. They can learn to use symbols such as Arabic digits. This has been taught both to chimpanzees and to monkeys, again, for instance, in Matsuzawa's lab. One can argue that there is a rudiment of culture in chimpanzees, for instance, because there is a process of local transmission of features that are shared by the local group but not shared across the whole species. But these cultural traits are quite limited, and they do not resemble the very large constructions that we see in human species. And particularly, there does not seem to be a process of cultural creativity, which is enormous in the human species, whereby we can play with our mental modules and discover completely new abilities that were not anticipated by evolution. So, this is, of course, an enormous question. What should be the biological foundations of human cultures? I'm going to try to ask these questions in the domain of reading and arithmetic, so narrower domains in which they begin to become possible to study. So what brain architectures support these cultural inventions of reading and arithmetic? What are the novel features of brain organization, if any, that make us the cultural species by excellence, the only species capable of cultural invention? Now, very often this question is not asked at all in the social sciences, and is not considered relevant, in fact, to ask about brain organization for culture, because one may say, I'm sorry if I caricature a little bit for the sake of time, but there is a standard social science model for which brain architecture is largely irrelevant when it comes to understanding our cultural acquisitions. Many social scientists would implicitly or even sometimes explicitly adhere to a view that may be called generalized brain plasticity or cultural relativism, and it's reminiscent of what Steve Pinker has written about the blank slate, which is the idea that basically we have a global ability to learn any domain of culture, however varied, however complex. And of course it is reminiscent of what John Locke wrote when he said, let us suppose the mind to be white paper, devoid of all characters, without any ideas. How comes it to be furnished? So free completely from the constraint of biology, the human brain, unlike that of any other animal species, would be capable of absorbing any form of culture. Well, I think you have guessed that this is not the view I'm going to defend, but of course I'm not alone in this respect, and there are many people who think that this is not the correct view. I'm just going to quote from a very respectable local person, Noam Chomsky, in Knowledge of Language, who first summarizes this view. According to a commonly held view, it is the richness and specificity of instincts of animals that accounts for their remarkable achievements in subdomains and lack of ability in others, so they are human friends, whereas humans, lacking such articulated instinctual structure, are free to think, speak, discover, and understand without such units. Both the logic of the problem and what we are now coming to understand suggest that this is not the correct way to identify the position of humans in the animal world. But what is the correct way to identify the position of humans in the animal world? In fact, there is a lot of resistance, even from inside our fields in neuroscience and in cognitive psychology, to the idea that brain organization can constrain cultural acquisition. So is the brain even relevant? For some neurobiologists who defend a more constructivist line of research, such as Quartz and Sejnowski, for instance, the developing cortex is largely equipotential, free of domain-specific structure. So if this is the case, although we believe that psychological representation have a cerebral substrate, the cerebral substrate is largely irrelevant, but it's not constraining. In the opposite direction, line, who might say with Johnson Laird that the physical nature of the brain places no constraints on the pattern observed. So again, even though there might be constraints, they are not coming from the brain. I would like to argue that this is not the correct view, and that we have to consider the problem in its complexity with a hierarchy of nested levels of description of this incredibly complex organ, which is the brain, with culture and education being possible at the very top here, made possible by a basic behavioral repertoire, which itself can be related to specific regions and circuits which themselves rely on cortical columns made of neurons, synapses, and receptors. And in fine, all of these levels should be related by what I would call bridging laws. Following Jean-Pierre Changeux, the idea is that we are not going to reduce one level to the other, but we should at least find the laws that relate one level to the other, and when we do so, we are going to find that there are strong constraints, that not all possible cultural objects can ride on top of this complex hierarchy which we have inherited from evolution. This is very general, so it's time I get a little bit more specific, and here is the research program I will try to defend in these 3 lectures for putting some neurons in culture, if you want. The first key idea, is a methodological idea, is that the non-invasive neuroimaging techniques are now allowing us to study in quite some detail the brain mechanisms that underlie cultural tools. Not all cultural tools, but the most simple of them, like Arabic digits and written words. When we study them for both reading and arithmetic, we're going to find that in spite of the cultural variability, there is reproducible and partially specialized brain regions that underlie this ability. And of course, this raises a paradox, because we might have expected that since evolution did not have enough time to adapt our brain architecture to these recent cultural objects, we should have found a lot of variability. Actually, I consider it a major discovery of neuroimaging techniques, starting with Michael Posner's work and Marcus Reichel and so on, that we can find reproducible circuits in all of your brains today that, for instance, are associated with reading. So how are we going to solve this paradox? Well, I will introduce what I call the neuronal recycling model. It's a very coarse idea at the moment, needs fleshing out, but here's the coarse idea, that the architecture of our primate brain, just like that of any other primate, is limited. It's not this space of infinite plasticity. However, it is laid under genetic control, but with the fringe of variability and plasticity. Itself, of course, this plasticity is under genetic control. The rules of plasticity are under genetic control. But they open up a space within which new cultural acquisitions are possible, and they are only possible in as much as they fit within this fringe of plasticity. So each cultural object will have to find its neuronal niche, otherwise it cannot be learned. And far from being a blank slate, our brain adapts to a given cultural environment by minimally reconverting or recycling some of its cerebral predispositions to a different use. So for each cultural object we have to ask, where does this come from? What are the brain circuits that are responsible for the ability, and what have they evolved for, and how are they changed, minimally reconverted to this new use? If we believe in this neuronal recycling model, there will be consequences, and I'll explore 2 of them. The first is that there should be numerous cultural invariants. If we look closely at cultures, we should find that in fact it's not this space of infinite variety. And in fact, even in domains that we should not expect, like the particular shape of letters, as I will show you today, there should be invariants that ultimately should be traced back to neuronal constraints. And the second point is about the acquisition of the system. We should find that the strengths and weaknesses of our brain architecture should ultimately determine the speed and ease of cultural learning. So in the end, the message should be relevant to education, because by identifying the circuits that are responsible for acquiring reading or acquiring arithmetic, we should be able to identify what is easy and what is harder for children, and what are the easiest paths to guide them to the final state of mastering a certain cultural object. Well, so here's the plan of the 3 talks then. In the first talk today, I'll talk about recycling the visual brain from reading, and basically I will describe to you some of the findings on the cerebral and neuronal organization of the visual word recognition system. This is speculative, but there is already quite a bit of data, and I will show how cross-cultural regularities in writing systems and in reading acquisition can be understood in the light of the organization of this reading system at the brain level. Wednesday, I'll talk about space, time, and number, cerebral foundations and mathematical intuitions. I'll try to look at the most basic of mathematical objects. I'll talk mostly about number and a little bit about space, and I'll try to show you that we begin to understand the neuronal foundations of these objects in such a way that we can reconstruct our behavior based on our knowledge of these neuronal circuits. But there is this very interesting question of how much this system is changed as we have the ability to map it onto symbols such as Arabic digits. Thursday will be the most speculative day, as I will try to understand what's different about the human species that allows us to have such cultural acquisitions in a way that no other primate species has. And I'll talk about the human Turing machine, the fact that we may have a special system, which I call the global workspace system, which is capable of top-down recruiting old circuits for new uses in a flexible way, thus implementing a rudimentary Turing machine. So today is about reading. And I would like to start by citing this beautiful quote by Nabokov, who draws our attention to the miracle of reading. We are absurdly accustomed to the miracle of a few written signs being able to contain immortal imagery, involutions of thought, new words with live people speaking, weeping, laughing. What if we awake one day, all of us, and find ourselves utterly unable to read? Now, I don't know if Nabokov knew, but such a case actually exists. It was described by Dehaene in 1892, and it's a very interesting reading if anybody likes a novel story. In October 1888, one of Dehaene's patients, Mr. C, described how suddenly he realized by waking up in the morning that he could no longer read a single word. And Dehaene studied this case and gave the first description of what is now known as pure alexia, with the following symptoms. After a lesion to the occipitotemporal cortex, the lesion only was studied after autopsy of the patient several years later, and it was discovered by Dehaene that it invades the left occipitotemporal cortex, as seen here when you look at the brain from underneath. And behaviorally, the symptoms are the following. The patient could not read a single word. He would look at the newspaper, and even the title of the newspaper he would not read. However, he is not blind, and object naming, face recognition can be completely preserved in such patients. Speech perception, production, and even writing can also be preserved. Writing is very interesting because the patient will write down on dictation words that 10 seconds later is no longer able to read. So this pointed for the first time to the fact that there must be specialization for reading in a very interesting way, separate from other visual functions and separate also from other language functions. Well, pure Alexa still exists to this day, and you can find cases that are extraordinarily similar to Dehaene's original description. And this is my collaborator, Laurent Cohen, who is a neurologist at the Salpêtrière Hospital, who has described many of these cases, studied them together. And if you study many such cases, then you can look at what is the critical site for impairment in these patients, and you can intersect the lesions of patients with alexia, and you can compare it to the lesions of patients without alexia. This is ongoing work, but already the lesions point to a small area in the left occipitotemporal sulcus, bordering a few different gyros here, that seems to be the peak of the intersection of impairments in these patients. What is very interesting, well, this converges from other analyses, particularly from Antonio Damasio, a little while ago, and other groups, and it corroborates very nicely with what we can find as brain imaging in normal subjects. So if you come into an MR machine in a few minutes now, we can readily image the circuits that is activated when you read a word, as compared to simple resting situations, to just see the whole circuit. And what you can see is that many areas are involved, but very prominently is featured this left occipitotemporal sulcus activation, which you can see in slice here in the left hemisphere, which will systematically activate just after presentation of a single word. So I'm going to talk mostly about this system. I just don't want you to forget that this is one area within a large system that is involved in reading. But this area seems to play a crucial role in linking the visual areas of the occipital pole here, the occipitotemporal cortex, to the rest of the language system. So it is an interface, and perhaps the crucial bit in reading is that we learn to interface vision with the language system. We were quite lucky to be able to bridge this data together to look at the causality and also the timing of brain activations during reading in a special case which was published last year by Raphael Gaillard and Cohen and colleagues. This is the case of a patient who was studied because he was epileptic, and in order to study his case, the surgeons inserted electrodes inside the cortex which monitor the source of epileptic crisis, but also allowed us to record and to correlate the recordings from these electrodes with brain imaging results. And the findings were that there is an electrode here sitting in the left occipital-temporal lobe. The hemisphere has been removed, so you can see it, which was responsive to the presentation of words at very much the same location that we find activation in normal subjects in our other studies. We could tell the latency of this activation on 170, 200 milliseconds or a little bit more. And we could also show with fMRI that this activation falls into a mosaic of visual areas. There is a certain form of specialization for words, maybe not complete specialization, but you can see that relative to a control of scrambled images, in this particular case, in this single patient, words activated this small area in the left occipital-temporal circuit here, quite close to this electrode that was responsive to words, and other types of visual stimuli, such as houses, faces, or tools, as has been described by other groups, activated reproducible areas that you can see in color here. So small activation for word in the midst of a larger mosaic for representing visual objects. Now, what came as a surprise to us is that the surgeons decided to operate the patient and to remove a small part which they believe was the source of epilepsy, which was just beneath the place where we had this electrode that responded to words. And of course, we warned the patient and the surgeons that this could result in pure ataxia. This was likely to result in reading deficit, but it was judged that the HDC was so severe that it was more important to relieve the patient who was making several fits every day. And as expected, the patient became alexic. And you can see that here in his reading times, he remained able to read, but extremely slowly, and with this letter-by-letter pattern of reading. So the reading time becomes a linear function of the number of letters in the word. This is his reading time before and after as a function of the number of letters. This points to an interesting feature of this area. With this area, you are able to recognize a string of letters in parallel. It's an important feature of this area, and when you lose it, you fall back onto a much slower serial reading strategy. We could do a comparison of fMRI before and after the lesion. You can see the lesion here in black, as pointed by this green arrow here, and reproduce this mosaic of activation. And you can see that the only element that disappears from the mosaic is the activation to visual words. It's not falling inside the lesion, so the cortex, the corresponding cortex is still there, but it seems to be deafferented selectively in a manner that leaves the other activation intact, and indeed the patient seems to remain able to recognize faces and houses and so on. But I think this is a nice case that puts together all these methodologies to show that this area on 200 milliseconds plays a causal role in our ability to recognize visual words. Now, can we go beyond that and understand what it's doing? There are many experiments. I will not provide you or bore you with a complete review, but quickly, this area responds more to words that respect orthography than to strings of letters that don't. So it responds better to real words than to consonant strings. Now, you might argue that this is not very well controlled because, of course, one is a word, the other is not at all a word. Much better study came recently from Jeff Binder's lab looking at the statistical regularities of orthography. And what he did was to vary in the domain of non-words, things that are really not readable here, but he varied the statistical regularities with a closer and closer approximation to English by varying the frequency of the pairs of letters in those strings. And you can see that the amount of activation correlates very beautifully with the frequency of the bigraphs, the pairs of letters, specifically in this area in lower areas in the brain after you correct for the reaction time of the subject. So this tells us that this area has been adapting to the particular orthography that the subject is mastering. It has incorporated a bit of the culture of the subject. It does something else. It creates an invariant representation of the word. So you are able to read words that are presented in uppercase, in lowercase, at different sizes and shapes. And we can show that this relates to that area. So a racial duration here, followed by another word which is presented for a longer duration. And psychologists have known for a long time that in this situation you can get subliminal timing. So although the subjects do not see this word because it's sandwiched between masks, the reaction time of the subject is faster in these black columns here when the two words match, as in this example, radio and radio, as opposed to when two different words are presented as prime and target. These effects, which, as you can see, generalize both across same case and different case presentation, indicate that the brain is able to extract an invariant representation. It's able to notice that this is the same word in spite of the radical change in the shape of the word. And we could relate that once again to the same area. This is the area that shows this effect. You can see this effect in fMRI activation. There was a decrease in activation when you repeat twice the same word, whether or not it's presented in the same case or in a different case. A pattern of activation which is only found in this case. We can go beyond that and specifically ask about priming for pairs of words that do not resemble each other in uppercase and in lowercase, but which are solely the result of a cultural convention. So if you take the word cow and cow here, you can see that there is visual similarity, so maybe this would be sufficient to cause priming by itself. But now when you look at the word on the right, rage versus rage, if you look at every single letter, the shapes are completely different in uppercase and lowercase. Any of them could have been the lowercase letter for the other. It's just a pure cultural convention. Nevertheless, this area incorporates this cultural convention and shows priming for this type of situation in which there is no visual similarity. So this area is not about visual similarity, it's about abstract orthography. It seems to be doing this function in many different cultures. And I'm just showing you one example here of the case of Japanese, where there are two different reading systems, kanji and kana, and both activate this area very strongly. But in fact, in a recent review by Chuck Perfetti, it was shown that all reading systems, whether it's Hebrew or English or French or Chinese, activate this area and rely on this area for reading. It doesn't mean that there are no differences, but there are several order differences. For instance, in this study, we found that the kanji tend to rely a little bit more on the right hemisphere and also on the left in a more anterior fusiform area. But overall, the circuit tends to be the same in different cultures, and furthermore, the function tends to be the same as well. We studied priming in kanji with the help of Kimiero Nakamura, priming in kanji and in kana, raising an interesting question. This is the same word, but of course it's depicted in completely different notations, kanji and kana, that represent either the whole word or represent the syllables of the word. And we asked, would this area know about this convergence, which is of course much more abstract than the convergence between uppercase and lowercase letters? Well, what we found was that behaviorally, you can get subliminal priming across two different notations, such as kanji and kana, but in terms of the brain activation pattern, now we get a differentiation. There are at least two different areas with quite separate functions. When you do kanji priming, you find that our visual word formula here continues to show repetition priming. So it must be encoded the orthography, in a certain sense, the organization of the visual stimulus of the kanji character. But it's not showing priming from kanji to kana, from kana to kanji. Another area is, and this is this lateral medial temporal area. So here we have evidence for a kind of hierarchical scheme of processing, and it narrows down a little bit what is the function of this visual word form area. It's dealing about the visual shape in an abstract form, but it's not dealing with the lexical identity or with the meaning, and probably this is more related to the meaning, because in another research, Joe Devlin has shown that you can get priming, for instance, for synonyms, sofa and couch in this area, suggesting this is at the level of meaning representation. Well, I could go on and show you more and more data. It's very clear that we are narrowing down now the problem of reading to the organization of specific areas, but how is this possible? The evidence suggests that all good readers activate a reproducible and restricted brain area, which is highly attuned to invariant visual word recognition, and I did not insist on that, but the localization is quite amazing. It's within 1 centimeter. In fact, the peak coordinates are within a few millimeters, even across studies from different laboratories and different cultures. Now, how is this possible, given that we didn't have time to evolve a specific system for reading? Why did we possess one? Well, this may be part of the answer. that we are, after all, primates. This area does not come from nowhere. The idea is that this part of the visual system has an evolutionary older role in object recognition, and that we recycle it for reading. As we recycle it, its capacities for elementary shape recognition, position and size invariants are already present and are reused in the domain of reading. And in particular, case invariants can be understood as a kind of viewpoint invariant that we reconvert into the domain of reading. So let me review very briefly what we know about this area from neurophysiological studies, not from my lab, of course. From Brodmann mouse, we get the idea that there is a homology at a rough level between the organization of the inferotemporal, occipital-temporal cortex, which is involved in object, face, and written word recognition in humans, and the similar area of inferotemporal cortex in the monkey, in the macaque brain. Of course, there are many, many differences, but still we can try to understand the human brain from recordings in this area. And recordings indicate that there is an enormous amount of selectivity for particular objects. In this beautiful example from Tamura and Tanaka, you have a neuron that responds only to this chair, to nothing else. These neurons show plasticity. So it's not just that they respond to objects in a pre-wired way, but they can learn to respond to these fractal shapes, as shown beautifully by this work of Miyashita and colleagues here. And not only that, but they can learn to pair these shapes. So in this example, these shapes came by pairs, and the same neuron ended up responding identically to shapes that were paired with each other during the experiment. For me, this is reminiscent of the shapes of uppercase and lowercase letters. There's clearly an ability to associate completely random shapes with one another in the same cells. Now, these cells, in fact, rely on the recognition of smaller parts. And we might call this an alphabet of object features. Now, this is my terminology, maybe it's a little bit exaggerated, but it's to convey to you the point. The original studies by Tanaka and colleagues have shown that if you start from object selectivity, say you find a cell that seems to react to the back of the experimenter here in these blues, you can in fact simplify the shape that the cell is sensitive to until you find there is a minimal shape that is sufficient to make the cell fire just as much. And as you follow this simplification procedure, you always find relatively simple features that are sufficient to make the cells fire, that are organized in the surface of the inferotemporal cortex. What is most surprising to me is that this alphabet of shapes incorporates some of the shapes that we have selected as letters. So you can see, for instance, that there is something that looks a little bit like the shape of an epsilon or an E, there is maybe an 8 here, and so on and so forth. There is the beginning of a combinatorial alphabet for recognizing objects already in the monkey brain. Why these shapes exist is a matter of research at the moment, but one strong possibility was suggested by Irving Biederman that these shapes may be appropriate to make an invariant code for object recognition. The idea is that these may be non-accidental properties that remain the same as the object is rotated or gets more distance. And indeed, Biederman showed that if you delete this property from the drawing of an object, it becomes extremely difficult to recognize the object. Whereas if you keep exactly the same amount of contour, but you also keep these junctions that make non-accidental properties, the object remains recognizable. So maybe our inferotemporal cortex has learned either through its evolution or through its early ontogeny that these are shapes that are extremely useful for object recognition. And complex objects seem to be recognized by a combination of such cells. As shown by the work of Kamifuji and colleagues, the shape of a fire extinguisher, for instance, will activate a number of patches on the inferotemporal cortex, each of which seem to be reacting to a specific subpart of the object. For instance, the hose, or the handle, or the body of the fire extinguisher. And the combination is also represented by its own combination of cells. So we reach a model, and of course this is all work in progress and certainly not work from my lab, but there begins to be models of this hierarchy of inferotemporal cortex representation that reach the level of being able to recognize particular faces or particular objects based on the idea that you can recognize the whole face based on its parts, such as for instance the eye region itself, which might be represented by the combination of subparts, which itself can be decomposed and so forth, and this matches nicely with the architecture of the visual cortex, with its cells, each detecting with growing receptive fields combinations of features at the lower level. So, speculatively, my colleagues and I have proposed a model of how the reading system might be organized in this way, and how we might change our macaque inferotemporal cortex, if you want, or our monkey inferotemporal cortex into a reading system. The idea is that we have a hierarchy of cells, each of which has a receptive field, and looks at the results of cells at the lower level. At the lowest level, of course, this is not changed. There is nothing special for reading here. It's detection of light and detection of bars. But as you combine bars, you begin to have the sensitivity to combinations that make curves or make little T-shapes. As you combine these at the higher level, you begin to have the ability to recognize letters, and this might be occurring somewhere in area B4. But of course, letters only in a specific font and in a specific case. But as you combine these, you begin to have the ability to recognize letters in an abstract case-independent way, and as you combine these, you may have the ability to recognize bigrams or pairs of letters, and as you combine bigrams, you begin to have the ability to have neurons that respond to primitives of words, such as prefixes, suffixes, or roots of words, and collectively, this can create a code that is specific for a given word, and at the same time invariant for a case and a particular location of the word. This is just a model. It's very likely to be false, and it will probably serve only as a target for further experimentation, but we can use it to try to see if it fits a little bit with the pattern of brain activation. So we have begun to do this with this hierarchy here. This is work done with Fabien Vaquier. We can present false font stimuli, or at the next higher level, we can begin to activate the letter detectors of the model, but we do so here at the lowest level with very infrequent letters that should be weakly represented in this system. Or we can present frequent letters, but we violate all of the bigrams that these letters make. So we purposely make it so that all of these bigrams do not exist in French or are very rare in French. At the next level, we can present frequent bigrams, but make sure that all of the quadrigrams made by 4 letters do not exist in French. And finally, at the next higher level, we can make sure that the quadrigrams themselves are frequent, and so this makes a very plausible French word, maybe the name of a drug or something. And this is a real French word. The idea is that as we move through this hierarchy, we should be able to see the wave of activation moving through the neuronal hierarchy, more and more anterior and with more and more activation. And this is very much what we have found. This is work that is submitted at the moment. So let me guide you through this image. What we do is we look at the activation to words, and even though it's mostly left lateralized, there is a little bit on the right as well, we normalize it to 1, so that you can see then what is the activation in the other conditions relative to this state. So what you see is a relative amount of activation, and you see that as we move down in the hierarchy, we get less and less activation, but we also can see it moving posteriorly, as if within this area, which we call the visual word form area, we now see an internal structure, which perhaps relates to this hierarchical coding scheme I was describing to you before. So we can see that there was before just one area, now is a gradient of activation, presumably linked to a hierarchy of detectors. You can see this a little bit better on this slide. Each of these histograms is the response to a particular spot along the occipitotemporal pathway in response to this hierarchy of 6 different conditions of stimulation. And you can see that in the back of the brain, all of these conditions give exactly identical activation, but as you move forward, you get a steeper and steeper gradient. This is not the case in the right hemisphere, where there is just a little bit of a gradient here, but nothing is steep. There is essentially a blocking of the activation evoked by objects that are, according to the model, not well represented by neurons in the occipitotemporal cortex. There are many other ways in which we can test this model. I'm not sure if I will have time to go through them all, but for instance, we can play with real words instead of presenting to you pseudo words. And these real words, we can rotate them, or we can space their letters, or we can shift them in the visual field. And all of these manipulations, according to the model, should eventually break down the neuronal responses in the inferotemporal cortex at a specific stage. From animal studies, we know, for instance, that the resistance to rotation of inferotemporal neurons is about 40 degrees, 50 degrees. So we can predict that at this level, the ventral reading system should stop. Well, we might predict that there should be letter-by-letter reading. It's not that you become unable to read, but you are slow and you have to work letter by letter, probably using shifts of attention to move along the string of letters. So the evidence that we found is very compatible with this idea of a critical threshold beyond which the ventral system is no longer sufficient to read. You can see this is the reaction time of the subject, and basically, as we begin to degrade this word in these three different ways, we get slight but non-significant or very weak slowing of reaction time, and suddenly at this threshold we get a very large increase in reaction time, and the different conditions correspond to the effect of the length of the word, 4, 5 or 6 letters. You can see that this will slow down the subject during this phase of letter-by-letter reading, but not within this region where the ventral system alone is able to recognize the word. And in fact, in fMRI, we find that this threshold corresponds to the deployment of a common system, which is in the bilateral parietal lobes, posterior parietal regions that are involved with shifting of attention, as well as amplification of the posterior part of the visual word form region in its equivalent of the right hemisphere at the level that we think is coding for single letters. So an amplification of the letter-by-letter reading system suggesting that the ventral system is no longer sufficient. Similar evidence from lesions. So if you have a lesion of the parietal lobe now, suppose you have a lesion of this letter-by-letter reading system, well, we should predict that at this level you should be suddenly totally unable to read. Your ventral system is no longer responding, and your dorsal system is lesioned and unable to implement the strategy of letter-by-letter reading. And this is very much what was found by Laurent Cohen in this study with a patient with bilateral parietal lesions. When the word was normal, the patient was able to read it. If it was vertical or inverted, she was not. And as the word was progressively rotated, we could see that there is a sharp threshold around 50 degrees at which suddenly the ventral system, which is intact, is no longer able to recognize the word. And this fits very nicely with the pattern of resistance to rotation of inferotemporal neurons in the monkey. Same for letter spacing. We should predict that with spacing, suddenly the binary neurons should not be able to respond because they have only a certain receptive field, and we know the receptive field grows by a factor of about 2 or 3 by each step, so we predicted that the binary neurons should begin to have problem around the spacing of 2, and this is very much what you see here. In the last 5 minutes, if that's okay, I will examine two predictions or two consequences of this neuronal recycling hypothesis. The first one concerns cultural variability. If this idea is correct, the brain did not evolve for reading. I think this part is very obvious. But it should be the contrary. The writing systems have evolved to be easily learnable by the brain because we cannot learn everything. We couldn't have learned any possible writing system. So this predicts that there should be very strong cross-cultural universals that should be present in writing systems, and they should ultimately be related to constraints of our brain circuitry. Well, we could think that the shapes of symbols in our alphabet are completely arbitrary and accidents of history. The classical history is that the shape of the letter A comes from the shape of an ox head, which of course was a favorite in prehistoric painting, was used in proto-Sinaitic writing as a small hieroglyph and then became the symbol for the first letter of the word Alf, which means ox, and therefore became our letter A. But is this just an accident of history? Well, to me, this historical process is also reminiscent of this simplification procedure which has been used by Tanaka and colleagues. And it may be that as we simplify these shapes, we keep precisely what is sufficient for them to be efficient to activate our ventral visual system. And evidence in that respect has been provided by Changizi and colleagues in recent studies that have looked cross-culturally at the shapes of letters and whether they have universal features. This paper I found extremely interesting. It's looking at what are the shapes of intersections of lines in characters throughout the world, not just our alphabet, but also Chinese characters, other alphabets, and so on and so forth. And what they find is that there is a universal frequency with which these particular topological intersections of strokes occur in all languages in the world, in all writing systems in the world. So, for instance, the shape of L or the shape of T in characters, or the cross and so on, they each occur with a frequency which is the same in different systems. You can see here that this is the ordering of the most frequent configurations that were found by changes in the What is most interesting is not just that there is a universal here, but that this universal can be related to the statistics of natural scenes. So if you count in natural scenes the patterns of intersections of contours of objects, you find the same statistics. It's very likely that our visual system, either phylogenetically or ontogenetically, has adapted to these statistical regularities. But it's very striking that our cultural systems have also adapted, perhaps second-hand, because our visual system was good at recognizing these particular shapes, then they were co-opted in our cultures. Second prediction. If this neuronal recycling idea is correct, then the difficulty of learning certain concepts or techniques should depend on the distance between the initial function and the new one. So as we recycle our inferotemporal cortex, we should sometimes benefit from its initial structure, but we should also sometimes suffer from its particular features which were not designed for learning to read. So plasticity and invariance for position are very advantageous for reading acquisition, but other features might be detrimental to cultural learning, and there might be something like a neuronal panda thumb, an inability or bizarre quirk of our design of occipital-temporal pathways, which shows that in fact this is neuronal recycling which is occurring. Well, I'm going to suggest that this sort of panda thumb, this non-advantageous design feature, might be found in the recognition of symmetry of objects. And in this, I'm following work that was done by Corvallis and colleagues many years ago. We seem to have evolved a symmetry mechanism that helps us to recognize faces and objects regardless of their orientation. So even though this is a completely different pattern of light and one that you've never seen before, you immediately see that it's the same face. And inferotemporal cortex neurons seem to spontaneously generalize to mirror images in the animal, as shown by this beautiful work by Nikos Yogotetis. If you train a monkey to recognize a certain shape, and the monkey only sees a certain view of that shape, when you then record the response of the neuron to the rotation of this bizarre paperclip shape, you will see that the neurons show a sudden peak of activation to the symmetrical view of that shape. So there is a suggestion that we have this system for symmetry recognition. We start with this system, maybe. It's present in our evolution, it's very useful, of course, to generalize our knowledge to new objects. But of course, you don't recognize that these are the same shapes. These are completely different words or pseudo-words. And there is a good possibility that this is detrimental, this property of symmetry generalization is detrimental to learning to read. I would like to suggest that when we see errors in young children of mirror image generalization, we are seeing an effect of neuronal recycling in the process. So in this nice example here, a young child has been told to write her name next to the dot, and when she doesn't have enough space, she readily writes her name in mirror image here and here. Whereas a child with 1 year older will find a way to start writing the word a little bit to the left so that she has enough space to write it from left to right. It seems that we start in life with this amazing ability which has never been taught for mirror image generalization. And if you look at many children and if you use this task, there is a peak of being able to write in mirror image just as well as in normal image, and then the ability to write in mirror image decreases in the course of learning to read. So we've started to look at this with fMRI. This is pilot data, it's not completely finished study, but the way we look at this is we look for priming again, repetition suppression, when we repeat the same word, like piano and piano here, compared to a situation where we don't repeat the same word, or we can also repeat images, and we can repeat them in mirror image, so the first one is in mirror image here, or we can repeat the pictures in mirror image. And the prediction is that we might see mirror image generalization for pictures, but in adults we should have lost the ability for mirror image generalization for reading, and this is exactly what we find. First, the priming is not exactly the same location, consistent with the form of specialization of this visual word form system. Here, this is just repetition priming. But then when we look in this particular area, and we look at the shape of the response for pictures or for words, this is the priming for normal images or for mirror images. So we can see that there is essentially complete generalization to a mirror image in this voxel. But in the very same voxel, we have mirror image priming for words, we have repetition priming for words, but we have absolutely zero mirror image priming for words. So this, of course, does not prove that this has been unlearned. We would have to follow children in time in order to show that this is unlearned. But at least it's a proof that the very same voxel can have quite different response properties for two different domains, suggesting that there's a form of specialization, and we might have lost this ability for symmetry generalization. So I'm concluding now. I think that, of course, writing is a recent cultural invention. We didn't evolve for it. It does show a large degree of cultural variation, but it is not at all the furnishing of the mind's white paper. This is not the right view. We are able to read because we inherit from evolution an efficient object recognition system with enough plasticity to learn new shapes. And with the relevant connections, I did not talk about that, but I think this is an important point. It projects to the right areas, which are the language areas. And the reason why there is such a narrow system is that maybe not all of the occipital-temporal cortex is able to have the relevant patterns of projections. Cultural evolution in the domain of reading can be viewed as a slow discovery of the optimal stimulus for our occipital-temporal system. The system remains suboptimal, however, as attested by the example of neural synergy. Finally, if this view is correct, then we all learn to read with a similar brain system. There are now thousands of ways in different countries to learn to read, and therefore, this cognitive neuroscience data should be relevant eventually to the teaching of reading, and I'm sure that Galaburda will tell you about his studies of dyslexia, which I think are partially consistent with this story. Thank you very much.
Critics, but the fact of the matter is that I think both Marc and I are great admirers of Stan's work, and you can see why. The richness, as pointed out by Liz, of the experimentation and the theoretical background leading to the experiments is just unmatched. For those of us who've been dealing with the issue of pure alexia from the 60s, when I learned about that from Norman Geschrin, I mean, this explanation that he came to with Laurent Cohen implicating the word form area, it's a remarkably elegant one. In fact, it needed preliminary work to get to the word form area, because before that we actually had no motivation to reduce the lesion to its minimum important lesion, which involved that particular area that led to these patients reading letter by letter, but unable to read whole words. So, one example after another of this. Regarding some of the comments about could this be tabula rasa, obviously there's quite a bit of evidence from neurobiology, which is seldom brought upon to try to explain how unreasonable that hypothesis that babies have to learn reading from scratch using any part of the brain could be. The brain of the newborn already has multiple areas of high differentiation. They're present in the monkeys. To my knowledge, there are no areas that are present in the human brain at the architectonic level that are not already present in the monkeys. Now, in the monkey, of course, they're smaller, the neurons are smaller, the dendritic trees are smaller, they probably have less processing space, but in general, all of them are there. In fact, you can go back much earlier in development and show that before the cortex is actually formed, there are these patterning molecules that begin to establish the cortex into partial separate areas with different markers that will eventually attract different kinds of connectivity and make up different circuits. So a lot of these circuits are in place. So it's not for me very surprising as a student of both brain development and evolution to see that all these machines are already in place for learning particular kind of information. In the case of Stan's work, to learn a certain kind of writing system that fits with the physiological properties of the visual system. It's more difficult to know why is it that monkeys don't read, and I think that remains a very crucial question. Are the differences describable at the genetic level? There are genes that are expressed in human brains that are not expressed in monkeys. There are genes that are expressed in the left side of the brain that are not expressed in the right, and we know that to a large extent, Stan, except for a couple of exceptions, focuses on the left side of the brain. What's the right side of the brain doing? There's some reason to believe that the right side of the brain is more sensitive to variance, that it can read, but it has to read certain it learns to read a font, and that's all it can read. Could we expand this kind of research to see what are the reading abilities of the right hemisphere, for instance? It is quite interesting to suspect that you might be able to learn to read with the right side of the brain if you were willing to read always the same kind of font, the same size of print, that kind of thing. The right hemisphere may have slightly different properties. The other thing about Stan's explanation of reading is that it's highly visual. It is highly bottom-up. If you read his papers, he says it's going to be bottom-up. He wants to make it simpler. There's a lot to learn from the bottom-up percolating of visual information up to the point when you can actually do letter strings into words. But reading is a lot more than that. And it's very important to know how language actually interfaces with the visual stimulus, because we know that in language-based learning disabilities, some of these visual areas do not function very well, even though these individuals don't show any visual problems, they show language problems. So at what point do the language areas actually creep in top down and influence the activity of visual system in that very cross-modal way? And when I was starting out, monkeys weren't supposed to have any cross-modal integration. If a monkey felt a shape and then it was shown a shape that corresponded to the one it felt, it wasn't supposed to know. But actually, monkeys do know, and we've learned a lot about them. To what extent then has cross-modal integration also evolved in the human to the extent that you can actually map linguistic kinds of experiences onto visual experiences, if linguistic experiences were in the auditory modality? And could that be a reason why monkeys can't read? Because they don't have, even though they have cross-modal association capacity, they don't have the extent necessary. But this is a remarkable start of the process of reading bottom-up, and I am sure that if anybody can keep it going to try to make it interact with language, spatial abilities, the ability, the contribution of the parietal lobe to this process, for instance, the spatial arrangement of letters, it's going to be Stan and his team. So I'm delighted to see this work and to be able to comment on it. Thank you.
Well, thanks a lot. As usual, you point to where it hurts in a very soft way. And of course, this is to cure the patient. I know that. I am the patient. So two short replies. The first is about the role of the right hemisphere. I am the first one to be concerned because it so happens that when I scan myself, I happen to have language in the right hemisphere. So I would like to understand why. And I think an interesting possibility is that, as I said, we don't have a wired area that is designed for reading. So what we might have instead is a kind of priority list of regions that are more or less suited because of their visual properties, but also because of their projections to language areas for learning to read. This is a little bit how I think about the problem now. So we know, for instance, I didn't show that slide from the beautiful work of Hauser and Marc, that there are gradients of selectivity for foveal as opposed to lateral places in the visual field. And the visual word form area tends to fall in an area of foveal selectivity, as we should expect. So this is one source of bias. Maybe it's the case that there is a left hemisphere bias, as you say, from some genetic ability, maybe a bias for more categorical representation that groups together several views into one representation. We also know it's plastic, and it can change. That's why I like to talk about priority list, because if the visual word form area is not available in development, another area will take over. We had a beautiful case of this with Laurent Cohen, of a young girl who was operated surgically at the age of 4 to remove entirely the left occipitotemporal region, and she learned to read. She was a little slow in learning to read, but she learned to read. And then when we scanned her at the age of 11, there was right-sided activation at the exact same coordinates, flipped in the right hemisphere, but exactly where it should be, and not at all a completely broad activation of the right hemisphere, a narrow one. So I think there is a list of constraints that together constrain this area to be where it is. And the left hemisphere seems to be preferred, maybe just because of the shortness of the connections to the language system. That's one hypothesis. Now you asked about the connection to phonology, and I think that's absolutely crucial and neglected in the model I've shown you today. And so it's really is a place where we need to enhance our reflection here. The model I presented is very much inspired by this idea that it's all about visual statistics, that we learn to compute visual statistics about the inputs, that our inferotemporal cortex is designed for that. So it's not, of course, a blank slate view, it's a view that this system is designed to compute this type of statistics. But I don't think it's sufficient. And I think you're perfectly right that it's very likely that the units that are extracted in the visual system are precisely the units that are appropriate to map onto phonology. And it's quite plausibly learning which is guided top-down by what are the relevant units that need to be communicated to the phonological system first. And there are more and more now in computational neuroscience perspective, there are now models that consider not just the statistics of the input, but what are the neurons going to be used for? What kind of task in subsequent areas are they going to be useful? And when you consider that they have to be optimal in that respect, not just in optimizing the statistics of the inputs, but in optimizing the output for a certain task, you find that the neural code can change radically, as was shown in the work by Salinas, for instance. So I believe this must be the case for reading, and we might find, for instance, that among the possible bigrams in a string, those that correspond to graphemes, those that map onto phonemes, are going to be privileged, whereas other bigrams that may be just equally frequent, if they don't map onto phonemes, they might not be privileged. This is a hypothesis for further research. I believe the tools are there now to look at these issues, and we are going to be able to understand eventually what is the code in this area, and I'm sure you will be right that it will be heavily influenced by phonology as well as by visual factors.
I too, as Al said, am one of Stan's fans, and part of what I appreciate, which makes it so hard, is when you cross over to so many disciplines. But of course, as Stan knows, that's where you become most vulnerable too. I want to do three things tonight, and I want to do it from the perspective of what would an evolutionary biologist say about the work tonight, rather than talk about scientists. So one of the things that sucks about reading is you have to wear glasses. This is an aging thing, and I'm going to put mine on. So the 3 points that I want to address tonight is one, the issue of the speed of evolution and how fast things can move, with the idea being that it's been too quick to have this be kind of wired into the brain. So I want to talk about what the assumption is, I think, behind that comment. The second thing is the notion that came up tonight, which is things that were selected for, and the issue of how things can get tuned up. So something being selected for in terms of an evolutionary process as opposed to things being tuned up in development. And I want to contrast that with things that were selected for as opposed to things that are now used for. And then the third thing I want to briefly mention is the plasticity of the human brain, and raise the question about whether Stan actually thinks that there's something actually special about the plasticity of the human brain that's different. So Al made the point that all the structures that we see in animal brains, monkey brains in particular, I think you were speaking to, are there. But then the question is, they're there, but are they as plastic as in the human brain? So as far as the speed of evolution, this is less of a sort of challenge specific to you than I think a challenge in general, which is that we don't really know about how fast evolutionary processes can take things. There's been an assumption that because things are complicated in some way, it must have taken a very long time to get where we are. So here's just an example of something where I think it happened very quickly, and we actually know, and it's a challenge to, I think, an assumption. So some of you may know that in all domesticated animals, the size of the brain is significantly smaller than the wild type. That's just true of all domesticated animals. So in 1970, a Russian biologist by the name of Belayev decided to take the domestication process very seriously and take wild silver foxes and try to see if he could domesticate the animals and see what would happen through the domestication process. In approximately 25 generations or 30 years, he took a completely wild-type animal and got a completely domesticated fox that acted like a puppy dog. Now, the method was very simple. He took experimenters who would approach the wild types, and those individuals in the population who allowed the experimenter to come close, he would take those and breed them. And then he would run the permutation again. And basically through that process, he got ultimately, in about 30 years, foxes that were basically domesticated. But the extraordinary thing about it is that although the only thing he did was simply look at approach distance to select among the variation, what he got was a constellation of correlated traits that were actually quite complicated. So he showed a decrease in brain size. He showed a massive increase in serotonin in 30 years. Now, what the underlying architecture there, it's also changed, nobody's actually looked. But in 30 years, you take a complex mammal, fox, and you get a massive change in the brain. So that's one, I think, issue we have to deal with. But related to that, there's been some very recent data on human brain evolution that shows that within the last 15,000 years, there's been a 100 cc drop in brain volume in 15,000 years of human evolution. So now, whether the complexity of the structures you're talking about are of that kind, I think is an open question. But I would like to leave it as an assumption that we don't really know about the pace of evolution, especially when there's targeted selection. So domestication is artificial selection. The extent to which natural selection can make similar moves when it's targeted, I think is unclear. The second point I wanted to make was about things that are selected for as opposed to things that are now used for. And I think the elegant argument, which I very much like, is that the structures in the brains that we see in macaques that evolve for general object recognition may now subserve a very different function. So the idea of recycling. I think that idea holds great appeal for biology writ large. So in a very nice recent book by Marc and John Gerhardt, two systems biologists, called The Plausibility of Life, they appeal to basic molecular systems where there's no real new inventions. There's just the reuse of different combinations in different ways. And I think the brain and lots of other parts of the natural world may appeal to that kind of argument. This seems to me, it's very much similar to the kinds of debates that people like Nancy Camisher have had about the nature of the face processing system and whether it's actually specific for faces. And I think this is where it gets to me to be difficult to actually resolve, because what you've got is an argument for the original function of something. And what you can say is that animals aren't reading, but we don't know what the actual original function is. So if the function is just generally detecting certain kinds of features, that may be even more ancient than the macaques. In fact, there may be other structures in their brains that do very similar things. So here's an example. There's some beautiful work on jumping spiders, so we'll move taxonomically about as far back as we can go. And it turns out that jumping spiders, when a male courts a female, he does a display like this with his front appendages. And you can trigger a complete courtship response by just showing females a triangle that goes like this. Now, the question is, do you want to argue that that facial sense is that old? Because there's a triangle, a very clear, good form, right? But going back to spiders, completely processed in a different structure of the brain, probably very different, so not inherited from spiders. So I think generally responding to certain kinds of primitive forms may be really, really ancient. And then the question is, what did it evolve from, as opposed to what it's currently used for? The third point, and I think I want to pick up on a point that Al made here and tie together, is that is the apparently exceptional plasticity of the human brain. And I think one way, which is extremely interesting, and you and I were talking about this a little bit before the talk tonight, is all animals communicate, but we are the only species to communicate via language, which is completely amodal. Any modality, well, not any modality, but many modalities will work. And that's just not true of any other species. So if you take a songbird, you knock out its syrinx, which is where it produces the source of sound, it's finished. There's no more communication to be had because it's very impoverished visually. If you take a primate and you knock out its facial features, which they've done, so they've done stuff that they've denervated the facial nerves, and the animal's now flat facially. They don't do something different to accommodate for the gestures and signals that were then operated in the facial domain. So we have a plasticity that's modality plastic that seems to be unprecedented, even though there's cross-modal integration at some level. So it's the question of, given that you can do cross-modal integration, why don't you integrate that into an output system which is communicative, given that the modalities are talking to each other? So I guess the question I'd like to leave off with is, what is it about the brain, the human brain, which not only allows plasticity for change and the kind of rapid tuning up that you can see, but also for the complete cross-integration of different systems that allows for productive output that you don't see in other animals, even though those structures are there.
Thank you, Marc. These are excellent remarks. As far as the speed of evolution is concerned, although I share your concern that it's difficult to measure and evaluate, I think in the case of reading we are on very safe ground for many different reasons. One is that the system is really recent from a cultural point of view. So the oldest date is 5,400 years, but that's only for a specific system. You have to realize that just a few centuries ago, most of the population was not reading at all, so there was no selective pressure to evolve any specific reading system. In addition, it seems to be the case that the more you read, the less you reproduce. So I think we are on safe ground. Of course, the analogy to domestication is very interesting. In a certain sense, we are domesticated animals, but it's not just going to be about brain size if you want to account for this type of very complex traits. Now, about the issue of primitive forms, I think that's a very interesting speculation that in fact the ability to recognize primitive forms is a very old feature in evolution, and I am ready to take it as a possibility that what changes he has discovered, the universal topological features that are extremely regular in the natural world, might be extracted by very simple species because they are so useful for describing the external world. So in that sense, I would not be surprised, but I think this would only enhance my argument if we found that the traits of our cultures in reading have in fact a much longer history that in fact relates to the very ability to survive in a certain structured visual world. And in the case of arithmetic, tomorrow I make this argument that in fact there is a very, very long history of evolution of the system, and what we call mathematics is based on a very ancient system, a foundation system for number. You're absolutely right that the idea of recycling is, in a certain sense, the idea of exaptation, which is present throughout biology. I like the idea of giving it a special name because I think there are special features associated with cultural recycling. One of them is the speed of this process, which does not require any change in the genome, of course, but is all going on in the course of the education of a child or adult. And it's absolutely true that there is a much greater period of plasticity, this was also mentioned by Al, in the human species compared to other species. That has got to be a feature that is essential in the ability of humans to develop cultural systems. We have this extended period of plasticity, and we take advantage of this, obviously, in our education systems. So it's probably no accident that we teach reading at a specific age, which is relatively young, and it would be probably much more difficult for our visual system to adapt if it was done at another age. But this is all speculation, and I would rather stick to experiments to investigate that. We are going to do new experiments now, and I'm excited about that, to look at what happens in illiterates, both as they are scanned while they still cannot read, and after a period of alphabetization, when they leave alphabetization school, they can read. They are not perfect readers, but they have learned to read, and we will be able to compare the brain organization and perhaps see what is the function of the specific area before these individuals learn to read and how it is changed. I think this is the kind of research that needs to be done. I think I sort of answered your criticism. Maybe we should open it up
So
for
why don't we
discussion.
open up to questions and why don't you feel that?
So, who would like to ask a question? Yes.
Relating to your pre-classification of Jankizi and Shimoto as the sort of universal statistics of graphing, based on that, if that idea is correct, one would predict that you should be able to design graphemes that would be very difficult for the system to learn. Yes, I'm wondering if you pursued that.
Not personally, but I think that could be an interesting line of research. People have worked on artificial orthographies, but mostly they've tried to design by hand orthographies that are learnable. I'm sure there are plenty that are very difficult to learn, and as an example, you might consider, for instance, the barcode, which we are confronted with routinely, but is almost impossible for us to decipher.
The flip side would be you could maybe tweak existing systems to make them easier to learn.
Yes.
Has that been?
That's a very interesting possibility. I am afraid that the resistance of cultural systems that are distributed amongst millions of individuals will prevent this type of application of cognitive science, I'm afraid. We have to be a little bit realistic, because in France we were not able to remove, after years of discussion, the circumflex accent, which is bugging us. So I believe that it will be quite difficult to change. But if you look at the organization of the alphabet, it certainly is not optimal. The presence of letters that are differentiated by mirror symmetry is clearly a suboptimal feature that creates some problem for children. It's interesting that some of the early alphabets could be read in both directions. So ancient Greek, for instance, could be read in what is called boustropheden. So you read one line in one direction, and you reach the end, and you read the other line in the other direction, and the whole word would flip. The same for hieroglyphs. If you look at hieroglyphs in temples, you see that for reasons of symmetry, often the same writing can be written on one side in one direction, the other side in the other direction. This is a feature that we have lost. And I'm not exactly sure why, because it seems that this would fit better of a visual system. But there are probably other constraints that come also from the writing. From the point of view of writing, maybe it's faster to run a fixed plan from left to right, for instance. All of this is, of course, extremely speculative, but could be addressed by artificial alphabet experiments, just like you said. Yes?
So, despite the fact that you have a French accent, I can still understand everything you said.
Sorry.
I'm just curious whether you had any opinions about whether you could test your recycling hypothesis to the auditory cortex and the recycling of, for example, the auditory recognition and auditory scenes.
Well, that's an interesting possibility. I haven't thought too much about that. I think the recycling model has some generality. But are you thinking of a specific example of an auditory ability that we are
learning? You could test the specific generalities in languages. The same is true for languages like mother and father, papa, baba, it's the same across many languages. I was wondering if you thought much about that.
I think a major The case of the number system is an interesting situation. Tomorrow I will describe some of our studies in Amazon, where we find that there are still a number of people who use a very rudimentary number system with just a few words, up to, in some cases, 2, in some cases 5. And it seems that the invention of a syntax of numbers is a late invention, which has a whole history derivative from the use of the body parts to refer to numbers and so on and so forth. So certainly in the small world of the syntax of numbers, I think an argument like what you would like to make can be made, that this is a process of cultural evolution. Whether this holds at large for other features of the language system, I really have no idea. Yes?
If I understood you correctly, you were arguing that the language processing basically builds on the lower complexity of the visual processing forms and then evolves to higher ones, right? It moves forward in the cortex. Now I wonder if you take a language or if you take a simple system like kanji, which has an enormous range of characters, whether you can correlate the order of learning the kanji, like in their complexity with respect to using these systems, with sort of a step-by-step development of first learning simpler forms before being able to learn a more complex form and then use it, and if that can be observed.
I don't know. I think the process is probably more complicated than that, because in fact the order in which you learn the kanji is determined by the culture. There is a certain order in which the characters are taught, and I don't think it's just by the simplicity of the character. But in a certain sense, what you are saying might be true. There must be a hierarchy of visual organization similar to what I described for the alphabetic form, which may occur also in Chinese or in Japanese characters. And in fact, it's reassuring to see that Chinese psycholinguists who look at written language are developing models in which the whole Chinese character is not recognized all at once, not holistically, as sometimes we conceive that this was the case, but in fact it's recognized by subparts. And we might have neglected the role of subparts of the Chinese character in the recognition process. So they are finding, for instance, that you can get priming by subparts of a Chinese character, just like you find priming by subparts of an alphabetic string. It's very likely that we have a similar hierarchy in the domain of other orthographies like Chinese or Japanese. It would be very interesting, of course, to flesh out a little bit more in some detail the kind of model I was proposing. It's just a model on paper, I should insist on that, this LCD model. I wish we had an actual computer simulation that could be fed with alphabetic inputs or with Chinese inputs, and we would see what kind of units emerge in this learning system, and we might make some better predictions as to what should be the organization of a Chinese reader or a French reader. But that I think is for the future. Liz, you want to?
Can I ask you a question that gets back to questions that both Al and Mark asked and that you raised in your talk, but I think I don't understand your answer yet. Here's two different ways of putting it. One, why are only humans doing reading? And two, why is reading so closely connected to language? From what you told us about these visual form recognition processes, it seems like it would be extremely useful to any creature with or without language to use abilities to recognize complex conjunctions of forms, to trace its way home so it can find a path on repeated occasions, to mark the location where some dangerous predator was found, to use visual symbols quite independently of language, so that a creature with no language or a creature like a human with language would use such a system without connecting it to language. Why do you think we see, one, only creatures with language having such a system, and two, our use of this system so tightly tied to language, both in where you find it in the brain and functionally?
I'm not sure if there is such a tight relation to language, but maybe we can start by listing what is probably not the reason why monkeys have not developed a simple system. They can learn these shapes, and they can learn to map them onto certain meanings. There are numerous examples. I showed the examples of learning factors or learning arbitrary paper clips, or I didn't show this, but the learning of arithmetic Arabic symbols is really amazing. The work of Matsuzawa really shows that chimpanzee, but there is also work in monkeys, really can recognize very quickly the shapes of Arabic numerals and know their meaning. So it's not, I don't think it's a matter of learning, except perhaps in the size of the repertoire, which can be there, which might relate to the size of the brain perhaps, but there is something more, which is, I think, the ability to invent these systems. And in order to invent these systems, I anticipate on the third talk now, which is more speculative, but I think you need to be able to play with your modules in new ways, so to try out the design of the new stimulation for a visual processor. So something that animals essentially never do. And the second thing you have to try to do is put together two processors in new ways. Find flexible linkages that you can try and let go if they don't work very well, or stick to them and train them actively if you find that there is an interesting connection here. So I would suggest that the reading was developed because we played with our visual system. We played with the idea of stimulating it with strokes on sand or stroke on clay, and we found then that surprisingly it could stimulate another processor. And we had the idea that maybe we could map these two processors together, the semantics processor or the language processor and the visual processor. I don't think that language plays a specific role in that respect. I think that we do that with all sorts of our mind's processors, whether they are linguistic or not. And I would rather seek the answer in our general ability to have a fluid way to map even non-linguistically. But I think you disagree on this point. I think you think that language plays a special role.
We'll take one more.
One more, yes?
Accepted that there clearly is an area which is indispensable for word-form recognition. This is what you imagine the neurons of that area contribute to word-form recognition, and more specifically, rather in the sense of Carl Wernicke, are the agnostic cells are the most necessary and sufficient for encoding or representing some prime number of word-form representation.
I think that when it comes to adults, the role of these areas become extremely specified and important in themselves. So the cases of pure alexia that I have studied with Laurent Cohen and that he has studied, it's quite rare that they recover in any very important way. I don't know if you would confirm that, but many of these patients can remain for years extremely desiring to be able to read, trying to teach themselves to read, but it seems that the level of plasticity in the adult brain is such that they never recover or relearn like a child. So in that sense, I would say that yes, there are very indispensable circuits, and within those circuits there are very indispensable neurons, and each of them, according to the kind of model I'm proposing, has a very specific role. They are very little gnostic cells. It's very speculative, of course, but the type of model I like to propose tries to anticipate on the time when we will be able to record from these neurons. At the moment, all we have is fMRI and some intracranial work, which in my case is just local field potentials, so we cannot see the single neurons. But if this kind of theory is guiding us, maybe we will, when we are able to record from these neurons, we will sort of know the right stimulus set. And if I am right, we should find neurons that respond to prefixes, for instance, or specific prefix, or specific suffix, or specific word root, and we should find that they make a combinatorial code and that are quite specifically located. I stick my neck out, as you can see. None of this data exists at the moment, but it's close enough. So people like Isaac Fried here in the US, for instance, have begun to make for a number of years now single neuron recordings in humans. And they find gnostic cells that respond just to the face of Bill Clinton, for instance. That's an actual case. So I think we will have these answers, and we will have them in 5 years, 10 years. Thank you very much.
Thanks, Dan, and have a great back.